Hello and welcome to the short 20-minute wrap-up of a lecture on basically organic polymers. Now what we're going to do is we're going to be talking about these things that we call condensation reactions and dehydration, or specifically dehydration reactions, and also hydrolysis, which are the reactions that either make or break down a polymer. These are, again, afforded to us by enzymes, which are macromolecules that speed up this particular process. Again, what we're going to see is that we have a short polymer, which is many of something being put together, and then we have a monomer, and then we'll see a release of a water molecule, and then the formation of a bond, or sharing a covalent bond between that monomer and a growing polymer. Again, that's really important, is that we know the orientation that this polymer is going to work, and so this can give off hydroxyl group. So that is what we're going to talk about. In, again, hydrolysis, water breaking apart, we see where a water molecule is introduced into those two molecules, into that one molecule to break a bond. We would protonate one side and we would put a hydroxyl group on the other side, and that would release the monomer for use elsewhere. Not going to talk about transfer right now. Again, each cell has thousands of different types of polymers, and those polymers are made up by relatively simple molecules. Again, the carbohydrates are the first polymer that we're going to look at, and basically they are made up of monosaccharides, or simple sugars, or single sugars. Mono meaning one, saccharides meaning sugars. The carbohydrates are macromolecules, are polysaccharides. They're polymers composed of many sugar blocks. And again, the monosaccharides have simple molecular formula, typically in multiples of CH2O, hence the name carbohydrates. Glucose, C6H12O6, is the most common monosaccharide that we'll be looking at and looking at the biochemistry of. But you need to be aware that there are other hexosucroses that are just as important, are hexoses. And again, an example of that would be glucose and galactose are very similar to one another, and fructose is very similar to one another. Again, they are both 6-carbon sugars, but the fructose actually has this structure right here, and the hexoses have a slightly different structure. And we also see the results of that structure when we look at the Benedict's reaction. Again, we are going to think of sugars as these circular molecules, although they flip back and forth from linear forms to ring forms, but we're not going to get into that. Disaccharide is formed when a dehydration reaction joins two monosaccharides, and that's what we call a glycosidic linkage. There's a number of different ones. Maltose is the glycosidic linkage, a 1,4 glycosidic linkage between maltose and glucose. And so we have, here's the 1 sugar here, here's the 4 sugar here, 1 sugar here, 4 sugar here, and we have that 1,4 linkage. Again, in fructose we have the 1, 2, in glucose we have the 1, 2, and because we have that 1, 2 glycosidic linkage, we are actually going to lose the ability of this molecule to react with Benedict's reagent. You probably saw that in class. Polysaccharides are polymers of sugars. They play a storage and a structural role. Starch is a storage molecule. Again, in plants we have amylose, amylopectin, which can be branched forms, and in animals we only see glycogen, which is multiple branched forms. Again, glycogen is the storage polysaccharide in animals. Cellulose is a major component of tough cell wall of other plants, and because again we have two ring forms of glucose, the alpha and beta. I'm not going to get into those too much, but basically a 1,4 linkage of the glucose monomer gives us hydroxyl groups all on one side, and so this molecule will actually form a kind of helix-like structure, rod-like structure. Say, well, that's going to be very strong, but it's not, because it allows for actually molecules to come in here and attack it on either one. Whereas the cellulose 1,4 linkage, again, because we have those hydroxyl groups on kind of opposite ends, rather than forming again bonds within itself, it forms more readily forms bond with another strand. And so again, that gives us a structure that looks a lot like this. Because these strands are so close to one another and they're bound here, basically means that we're excluding a water molecule from going inside of there. Again, the only way to digest these, and no vertebrates can digest these, but what cows do is they actually have trained some protozoans and some bacteria to work on breaking down the starch, sorry, not the starch, but the cellulose molecules. But again, their teeth play a very important role in breaking down those fibers. We're not going to get into that, but there's other structures. Chitin is a very similar. Lipids are the large biological molecule, but they are not necessarily polymers, or they're very small polymers. Fats are made up of two components, and we see a glycerol, and those are bound with a fatty acid. Fatty acid has basically a carboxyl unit over here, which would be hydrophilic, but it has this very large hydrophobic region over here. Again, when we look at fats, is triglycerol. We have saturated fatty acids and unsaturated fatty acids. And so saturated fatty acids are normally the organisms that can withstand a higher temperature. Sorry about that. And we tend to see these more in animals. That's the reason why butter is solid at room temperature. And then plants and some fish have unsaturated fatty acids, which allows them to be more liquid at kind of lower temperatures. Phospholipids is a very special type of lipid where we have a hydrophobic head. If you remember, in the fatty acids, you kind of lose that acid group on the end here when you bind to the glycerol. However, in the glycerol, we have a phosphate, which has a negative charge, and we have a choline. And so these phospholipids have a phosphate, and they also have an amino group inside of them. This gives us a hydrophilic, water-loving head and our hydrophobic, water-hating tail. Now, how will this self-assemble? With very little energy, if we put a phospholipid in a liquid, they're going to self-assemble into these things called a phospholipid bilayer, where we have the hydrophobic region reaching water on this side and the hydrophobic region reaching water on this side, and then where all of those tails will interact with one another. And again, a phospholipid is an example of an amphipathic molecule. Okay, and we will learn more about these when we look at lipids. Steroids and cholesterol are again formed by kind of flat rings that interact into the fatty acids. They make them more soluble, and they give you a little bit of structures. Proteins are responsible for 50% of the dry mass of the cells. Again, this table here, an overview of kind of what the cells, what proteins do. And one of the main things that we study proteins is because they are enzymatic. But again, they play a very large role in structure, in storage, in transport, hormone receptors. So again, proteins aren't just enzyme anymore. It's not one enzyme, one gene, as if you've kind of heard that in the past. The enzymes are, again, a type of protein that acts as a catalyst, and so what we see is the protein is able to kind of temporarily bind to the substrate in order to produce some strain on the bond and either break the bond or release the bond. In this case here, it doesn't show it, but we have a water molecule needed to come in here, and that water molecule is used up in order to make these new structures. A polypeptide is a polymer built from the same set of 20 amino acids. I really think that proteins are the little nano robots of our future, and that we're going to use them more and more as we're able to build more of those. But basically, a protein consists of more amino acids. Know the basic structure of, again, an amino acids. We have this R group, we have an alpha carbon that's attached to a carboxyl group, and an amino group. Okay. Again, have a general idea of what the different amino acids are out there. There's several of them that we use all the time, like glycine, like phenylalanine, like cysteine, that's able to form the disulfide group. We have acidic and basic ones. So basically, if I gave you an amino acid R group, you should be able to tell me kind of what the properties of are. I mean, again, is it polar? Does it have a charge? Is it non-polar? Okay, give me an example of that. The amino acids are linked together by what we call a peptide bond. It's a covalent bond, and so here we have the start of the amino group, and it's the carboxyl group. That's important. We have a loss of the hydroxyl group, and we loss of the hydrogen group from the incoming monomer, and that releases a water. So this is a dehydration synthesis. We went from a dimer to a trimer. Okay, and again, this is the backbone, and these are the side chains. So a lot of times when we look at it, this is what we call a primary structure, which is the sequence of which amino acid comes after which amino acids. These amino acids in their line can be twisted and folded into various forms, and we have several ways of looking at them. In this case here, we're using a ribbon model or a ribbon diagram in order to look at the twists and the folds that make up a protein. But again, remember, the protein's main structure is due to those valence electrons on the outer surface. And so the outer surface, or specifically areas like this, like a groove, are very important in the binding to the substrates. Okay. Again, why that is important, in this one here we have a combination of both of those. This is an old model, but it's really cool. An antibody is a protein that's able to bind to specific molecules. It can bind to a flu virus or flu protein. And how it does that is it has a compensatory shape. So if we look at the shape of the protein, it's shaped something like this. If we take a look at this molecule, it's shaped in the matching area. And what you don't see is that there may be a charge over here, but there's the equal and opposite charge over here. There may be some polarity over here that we see over here. There's some hydrophobicity over here that we see matching over here. And so a little change, like the change of this positive group to a negative group, can greatly affect how a protein, or in this case here an antibody, will bind to something like a virus. The primary structure is the sequence of the amino acids, and so that's this structure right here. Secondary structure is basically how that sequence kind of folds in on itself, whether it's a helix or that. And tertiary structure is basically the association between those secondary structures, and the quaternary structure is an association of peptides. And so we'll take a look at that. The secondary structure, again, that helix. Again, we have hydrogen bonding between those. I can't find my hydrogen bondings between these structures right here, and the hydrogen bondings within the different lanes gives us that structure. And it's the tertiary, quaternary structure is basically due to things like hydrophobic interactions, hydrogen bonds, disulfide bonds. So we have this long structure, and this gives us some of the properties of proteins. And so if we have very long, solid peptide chains, you know, this looks like a girder. This looks like it's strong. Again, this here looks like it's meant to float around in our blood, which is hemoglobin, which will bind to oxygen. And quaternary structure is two or more of these together. Again, this folding of a protein can fold in very many ways. And so we can't just look at a sequence of a protein a lot of times and tell us what the most natural folding would be. Not yet. Hopefully we will do that. When we unfold a protein due to breaking these intermolecular reactions, let's say adding heat to it or adding certain chemicals, etc., or acids or bases, we can unfold the protein, in its case what we call denatured. This protein is not active. And renaturation is that very unlikely, but sometimes we can fold that back in together into its normal shape, and the protein will be functional. There are a class of proteins, very interesting proteins called chaperonins, that are responsible for kind of helping maintain the shape. X-ray crystallography. I'm not going to get into that. And then nucleic acids is basically looking at the nucleotides. It's very important that you know the monomers, which are the nucleotides, and the polynucleotide gives the structure. And so we have a nucleoside, which is nitrogenous base that's attached to a sugar that's attached to a phosphate group. So we have base that's attached to the sugar. It's attached to the 1 carbon of the sugar. It's the 3 carbon that's and the 5 carbon that's attached to the phosphate, and so the 3 carbon is attached to the phosphate of the 5 carbon of the other one. Again, know the base pairing rules. Know the different structures of these guys. Oh God. And I am and basically this is the structure right here that you need to know what the structure of DNA looks like. And again, your book gives you a chart to know again what the name of the molecules are, what its components are. Give me examples and give me functions. So again, if I give you a function, you should be able to give me what a molecule that does that. If I gave you a component, you should tell me what that molecule is. And that's basically about it.